Plasmid purification. 19. Obtain 2 500 milliliter flasks containing transformed bacteria grown overnight from the shaking incubator. 20. You can prepare several glycerol stocks of transformed bacteria. Pipette 150 milliliters of glycerol into an Eppendorf tube and add 850 milliliters of overnight culture to the tube. Pipette up and down to gently mix. Place in a negative 80 degrees Celsius freezer. 21. Harvest the bacterial cells by centrifugation at 6,000 times G for 15 minutes at 4 degrees Celsius. Observe the bacterial pellets. 22. Pre-chill the neutralization buffer at 4 degrees Celsius. 23. Add RNase A and lyse blue to the resuspension buffer bottle to achieve a 1 to 1,000 dilution. Lyse blue is a color indicator that is added to provide visual identification of optimum buffer mixing to help ensure efficient cell lysis and precipitation of SDS, genomic DNA, and cell debris. 24. Resuspend the bacterial pellet in the Tris-CI EDTA resuspension buffer. 25. Add the lysis buffer to the bottle and mix thoroughly by vigorously inverting 4 to 6 times and incubate at room temperature for 5 minutes. Do not allow the lysis to proceed for longer than 5 minutes to prevent overexposure of the plasmid to the denaturing conditions which can affect the final plasmid yield. The lysis buffer contains sodium hydroxide and SDS, which solubilizes the phospholipid and protein components of the cell membrane, leading to lysis and release of the cell contents. Sodium hydroxide denatures the chromosomal and plasmid DNAs as well as proteins. The addition of RNase A ensures that cellular RNA is digested during lysis. Note, after the addition of the lysis buffer to the resuspended bacteria in the resuspension buffer, the color of the suspension changes to blue due to the addition of lyse blue to the resuspension buffer. Mixing should result in a homogeneously colored suspension. 26. The neutralization buffer contains potassium acetate. The high salt concentration causes potassium dodecyl sulfate to precipitate, and denatured proteins, chromosomal DNA, and cellular debris are co-precipitated in the insoluble salt detergent. Add the chilled neutralization buffer to the suspension, mix immediately and thoroughly by vigorously inverting 4 to 6 times. Note, upon addition of neutralization buffer, lyse blue turns colorless. The presence of a homogeneous solution with no traces of blue indicates that SDS from the lysis buffer has been effectively precipitated. Incubate on ice for 30 minutes. 27. To clear the lysate, centrifuge at 20,000 times G for 30 minutes at 4 degrees Celsius. Remove the supernatant containing the plasmid DNA promptly. 28. Centrifuge the supernatant again at 20,000 times G for 15 minutes at 4 degrees Celsius. Remove the supernatant containing plasmid DNA promptly. 29. Equilibrate a filter tip by adding the detergent equilibration buffer. 30. Apply the supernatant from the cleared lysate to the filter tip and allow it to enter the filter by gravity flow. Collect the waste filtrate in a plastic container. The plasmid DNA will bind to resin in the tip under appropriate low salt and pH conditions. 31. After all of the supernatant has been applied to the filter, wash the tip with the appropriate volume of the wash buffer to remove any contaminants in the plasmid DNA preparation. DNA, proteins, dyes, and any other impurities are removed by this medium salt wash. 32. Elute the plasmid DNA bound to the tip by adding the elution buffer, which provides high salt conditions to rebind the plasmid DNA from the tip. Be sure to collect the flow-through at this point containing the plasmid DNA. Collect the DNA in smaller centrifuge tubes. 33. Precipitate the DNA by adding the appropriate volume of isopropanol to the eluted DNA. Mix and centrifuge right away at 15,000 times G for 30 minutes at 4 degrees Celsius. 34. Decant the supernatant carefully. 35. Wash the DNA pellet with the appropriate volume of 70% ethanol and centrifuge at 15,000 times G for 10 minutes. 36. Decant the supernatant carefully. 37. Air dry the DNA pellet for 10 to 20 minutes and redissolve the DNA in an appropriate volume of buffer, such as TE buffer. 38. Transfer the redissolved plasmid DNA to an Eppendorf tube. 39. DNA concentration yielded from the procedure can be determined by UV spectrometry.